Okay, guys, chapter 26, part 2. Going to finish up electrolytes. And we're going to start on the acid base portion of the chapter. We're going to talk about one of the main electrolytes, sodium, major cation of ECF, that's AB1 stuff, causes one half of the osmotic pressure gradient, the other half, of course, being chloride. Between the ICF and the ECF, we actually only require between 1 to 2 millimoles per day. We actually eat about 130. This is why we have an epidemic of hypertension. We get rid of sodium mainly through the kidneys. Primary role in the context of this chapter is in controlling the ECF volume, the water distribution between the compartments. Of course, we also know that sodium has a big role to play in the action potentials in neural and muscular systems. Regulation linked to blood volume and pressure, which is what we said up there. The more sodium you have in your blood, which is going to equate with the sodium in the rest of the ECF, the more water you're going to draw in, the more plasma, the more you have, the more you have pressing on the walls, the higher the blood pressure. As far as our little table here, the ECF concentration is osmolality. We have osmoreceptors that report to the hypothalamus, which in turn is going to control the release of ADH from the posterior pituitary and stimulate the thirst centers in the medulla. Uh, Body sodium content, as we said, is related to blood volume and pressure. We have baroreceptors in the carotids and in the arch of the aorta that monitors blood pressure. These blood pressure itself is regulated by the renin, angiotensin, aldosterone axis, and also atrial natriuretic peptide. It's going to be a big part of that. Hyponatremia is just a lower level. I'm not requiring you guys to memorize these lab values because they fluctuate, but less than 135 milliequivalents per liter. Don't worry too much about the units. Just know that that's our lower level of sodium. How can we get rid of our sodium? Well, gastrointestinal suctioning, diarrhea, vomiting, diuretics. Some diuretics will cause it to pass out because we're going to prevent its reuptake in order to have the maximum amount of water going out to produce the most dilute urine. Inadequate salt intake. If you work in a very hot environment and you just drink pure water without electrolytes such as Gatorade, then you could do that. Low salt intake is not an American problem. And then we have a fluid shift from the ICF to the ECF. If you take something like mannitol, then by adding those extra solutes into the ECF, you're going to draw water from the ICF in order to dilute those. That additional water, if I add water and keep the amount of sodium constant, then I'm going to dilute it, and this is going to cause the hyponatremia. Some of the signs and symptoms: lethargy, headache, confusion, apprehension, seizures, and coma. What's happening? I'm also removing water from neural cells, guys. And this is part of the problem. And just so you know, if this is not treated, the last symptom, which is a permanent symptom, is death. Okay, hypernatremia. So if I have too much, 147, that's okay. That's a good number, Zanny. But I've got more than I need. How can I get it? If I get a hypertonic IV solution like 3% normal saline, although usually we only do that once when we're trying to correct hyponatremia, and even that is done very slowly. Oversecretion of aldosterone. Aldosterone is going to cause, if you recall, the reabsorption of sodium from the distal convoluted tubule and the secretion of potassium in its place. Loss of water. If you lose water but not sodium, if you lose more water than you do sodium, then obviously you're going to wind up with a higher concentration of sodium. Long-term sweating with chronic fever, you can lose more water. I know sweat is salty, guys, but you can lose more water than you do salt. Respiratory infection that leads to the water vapor loss. We breathe out pure water in our exhalations. If you have diabetes and have polyuria, you're going to lose a lot of water uh, because the excess glucose is going to keep the water in there. It's going to act as an osmotic solute. If you have insufficient intake of water, hypodipsia, that can do it. If you move water from the intercellular fluid to the extracellular fluid, it will dilute. And in that case, you're going to be dehydrating the cells, obviously. Hypernatremia, pituitary gland is going to release ADH. When the osmoreceptors in the hypothalamus are going to report this, and this is going to cause the release of ADH from the posterior pituitary. We're going to do this and we're going to reabsorb water. Remember, the ADH is going to put the aquaporins in the collecting duct and cause us to reabsorb water out of the collecting duct. And at the same time, we're also going to get aldosterone. We're going to reabsorb some sodium, but not much. Our sodium level is already high, so that's not going to be such a big thing. But we're going to reabsorb the water, and that's going to dilute it. Hyponatremia due to dehydrations, thirst, fever, dry mucous membranes, mouth, and could be the eyes too, depending. Low blood pressure, which will cause a reflex tachycardia. Low jugular venous pressure, restlessness, so forth. And you can get it by, they don't put it here, but you can also get it by excessive salt intake. Potato chips, popcorn, peanuts, that sort of thing. If you guys ever have had something really salty like that, you notice that right after that, you're going to get thirsty, and obviously you're going to actually have ADH increased, and the hypothalamus is also going to send messages to the thirst center and drive you to go and get a drink and take in excess fluid, more fluid rather, not excess. Potassium is a major intracellular cation. It's all there for the resting membrane potential. Doesn't really play a big role in osmotic balance. Secretion by the kidneys. Why do I put potassium inside the DCT to go out in the urine? If my potassium goes up, my potassium goes down, I'll put less. If I have a change in pH will actually cause more potassium to be out in the extracellular, or aldosterone levels. Aldosterone responds to potassium levels as well as to the water levels. Okay, effects of potassium imbalances. Normal kalemia, when we've got our potassium in the range, they don't put it down here. It's around 3.5 to 5.5, something around there. Again, depending on it, you don't have to know all this. So if I have the normal amount of potassium on the inside and the outside, then the resting membrane potential is going to be at its normal level. We remember that depolarization, I let sodium in, and then the sodium gates are going to close around the peak. The potassium gates are going to open, and I'm going to let potassium exit the cell. And by doing that, leaving those positive charges, I'm bringing it back down to the resting membrane potential. Now let's see what happens with hypo and hyperkalemia. Hyperkalemia is too much potassium. I'm going to have, so with hyperkalemia, we're actually going to raise the resting membrane potential. We're going to make the cell more excitable. How so? How is this going to be so? Well, let me draw it over here, guys. So this is the normal resting membrane potential, and this is the threshold that triggers an action potential. The more potassium I have in this area, then it's actually going to it's going to keep potassium from going out. The more potassium I have here, the more it's going to go inside the cell, potassium, sodium, potassium, the less is going to come out. It's going to be much more difficult to repolarize, but since I'm bringing more potassium in, I'm taking that, say, minus 30, and maybe I'm moving it to minus 15. I'm just picking numbers out of a hat, guys. But the point is, I've raised the raising membrane potential closer to threshold, so it's going to be a lot easier to get this guy to fire. May not be as easy to get him to repolarize, because repolarization is potassium leaving the cell. Hypokalemia, I have an extraordinarily low amount of potassium on the outside of the cell. Again, resting membrane potential threshold. What happens here is because there's less than normal potassium, I'm going to have an increase in the amount of potassium that goes out. So I'm actually going to lower the resting membrane potential. I'm going to actually hyperpolarize the cell. It's going to be much harder for it to reach threshold because I'm actually taking it, say, from negative 30 to, say, negative 40, for example. I'm taking it further down away from threshold. On the positive side, these cells are going to be less excitable because they have to have an extraordinarily large stimulus to reach threshold, but they should repolarize very quickly because there's so little potassium outside that the potassium will not have as much resistance. Hypokalemia, decreased intake, starvation, vomiting, diarrhea, alkalosis. Diabetics can also have this with the potassium. Insulin. Insulin is going to get the glucose to go in the cells, and the potassium is going to follow. Decreased levels in the blood, interstitial fluid, can cause we already talked about that, reducing neuron response to stimuli. Okay, so here we have a cell. In acidosis, I have an increase in the number of hydrogen ions right there. What happens is the cells are going to uptake the hydrogen ions, but in order to keep the electrical charge balanced, they're going to put out additional potassium. This is diabetic ketoacidosis. We have a shift like this. We bring the hydrogens in. The potassium artificially is inflated. If you correct the acidosis, we will actually be able to, the potassium will actually go back. So in alkalosis, we're going to have additional potassium or additional hydrogens leaving the cell, which means we're going to have to bring in more potassium. That's why alkalosis causes hypokalemia. Alkalosis, you're going to see shallow respirations, irritability, confusion, drowsiness. Your nervous system's not going to work right. Weakness, fatigue, muscles aren't going to do so good either, remember? Arrhythmia, irregular tachycardia, lethargy, a very thready pulse, very weak and fast. Decreased intestinal motility, you're going to have problems. Nausea, vomiting. Ileus is a paralysis of the small bowel. But constipation, nausea, vomiting, and ileus, because the smooth muscle is not going to work right. Neither are the nerves. Hyperkalemia, elevated potassium, increased dietary, which is going to cause you to have more ECF, partial depolarization or inability to repolarize. We talked about that, raising the resting membrane potential closer to threshold, skeletal muscle cells, neurons, and cardiac. This is a problem, guys. If you have too much, you're not going to be able to repolarize the heart cells, the cardiac muscle cells after contraction. That's not a good thing. That's why potassium, a form of potassium, is one of the drugs they use in their cocktail when they do capital punishment. So muscle twitches, cramps, paresthesias, low calcium can also cause cramps. Low calcium, low potassium, the Charlie horses, irritability, anxiety, decreased blood pressure, EKG changes, dysrhythmias, irregular rhythm, bad thing, abdominal cramping. You're going to have hyperexcitable cells. So the muscle cells are going to contract. And in diarrhea, chloride. Chlorine is one of the gases, very, very reactive, has seven electrons in its outer shell, so it's really reactive. This is going to be one of our main extracellular anions because it's paired with sodium. Water dissolves in, or salt dissolves in water. The ions that are attracted to each other, held together by electrical attractions, are moved apart because of the attraction of the water molecules around them. But this is the other half of that osmotic pressure gradient. Help us to maintain our hydration along with sodium, and balances the cations. So if I have a lot of sodium, and I do, then I need a lot of chloride. Excuse me, a lot of chloride to balance that. Otherwise, we'd have a voltage between different areas of the ECF, and that would be a bad thing. Bicarbonate, HCO3. This is a major anion, second most abundant after chloride. This guy is going to be involved in our acid-base balance. We're going to talk about that. We talked about this in respiratory when we talked about that most of this carbon dioxide that comes from the tissues, that carbon dioxide and water, there's an enzyme called carbonic anhydrase that's going to combine these into carbonic acid, H2CO3. Because this is an acid, one of the hydrogens is going to be easily detached. So it's going to break apart into an anion bicarbonate and a hydrogen ion. And this is the guy right there. We'll talk a lot more about this. I probably will just do it now. So CO2 plus water. Sorry guys, I still don't write very good with a mouse. Now notice that these arrows are going both ways. That means this reaction can go either way. And what's going to cause it to go one way or the other? Well, I'm glad you asked me because I'm going to tell you. And there it is broken apart into the hydrogen ions and the bicarb. I can run out of room. That's an O, by the way. There we go. This is what's driving it, guys. If I have too much hydrogen ions here, that's acidosis, then this thing is going to go this way. I'm going to take those excess hydrogens, bind them with bicarb, turn them into carbonic, and then that carbonic anhydrase, that enzyme, it's going to be right there, is going to break it down into CO2 and water. CO2 I breathe out, okay? That's what happens if I have too much. That's acidosis. Let me get rid of some of my stuff. What happens if I have too little hydrogen ions? That's alkalosis. Then this guy is going to go this way. I'm going to take CO2 and water, carbonic anhydrase, going to turn into carbonic acid. It's going to break apart, and I'm going to make hydrogen ions, or I'm going to release them. and that's going to increase it and bring it back to homeostasis. That's the key on the buffers. We're coming to the buffers. I just thought that was fortuitous time to mention that. Calcium, most of it is in our bones, a humongous amount. The calcium that's in the extracellular fluid is regulated by the parathyroid hormone. The parathyroid glands monitor the calcium levels. When they're too low, it's going to release PTH. When the PTH is going to go to the bones and turn the osteoclast on, who will start dissolving bone to release calcium back into the blood. The kidneys will also get, calcium is 2+ charges, by the way. The kidneys will also get the signal, and that will cause them to reabsorb more of the calcium in the nephrons. The small intestines will also take the vitamin D that was activated by the kidney and use it to reabsorb or to absorb more calcium. Also draw it like that from the intestines. The net result is we raise our calcium levels. When the calcium levels reach the optimum, PTH turns off. What happens if we have too much calcium? Calcitonin in animals, it does some good, but in humans, it does no good. It is a waste, and it does not help us. If you've got hypercalcemia, then you're going to have to get medical attention. Your body can handle low calcium to a degree, of course. Phosphate, 3 forms: dihydrogen phosphate, hydrogen phosphate, and then our phosphate group, PO4. All of these have a -1 charge. All of these have an excess electron, so that they're anions. Hydrogen phosphate is the most common. 85% of this is in our teeth and our bones. Phosphate along with calcium and hydroxyapatite crystals. Of course, we also have phospholipids in our cell membranes. ATP, of course, has 3 phosphate groups, 3 of these guys. The nucleotides have a phosphate group on them, and we also use some of these as buffers. They work kind of the same way. This HPO2 can pick up a second hydrogen ion, so the guys can go here. If it's acidosis, if it's alkalosis, these guys can go here. Okay. All right, guys, the tables. Sodium, potassium, phosphate, we've kind of talked about. Look over these things. I'm not going to read a table to you. That's it's boring for one thing. But look this over, and if you have any questions or anything you don't understand, oh, there's the levels. I guess I was right. I was guessing. In any case, don't worry about these numbers. You don't need to know them, but you do need to know that there is such thing as hypernatremia, hypo, and so forth. Some of the causes, we've gone over a lot of these. Some of the symptoms or the consequences, it would be good to read that. If you don't understand it, then you can let me know either in a class meeting online or email or just any way, and I'll be glad to explain it to you. Chloride, we talked about. Calcium, we need to talk about magnesium. Hypermagnesemia and hypomagnesemia. I actually had a case of hypomagnesemia in one of my ICU patients one time. Very interesting case. Yeah, alcoholic. Chronic diarrhea, severe malnutrition, too much diuretics can give you that. You can lose too much magnesium in the urine. And then the effects of it, I'm going to let you guys read that and let me know what you don't understand. Regulation of sodium. How do we control the sodium and potassium? Okay. DCT. Aldosterone causes us to reabsorb sodium. Sodium is a cation with a plus charge. If I reabsorb that plus charge, then I got to put a plus charge in there. The plus charge is potassium. Now, aldosterone. How do I get aldosterone? Well, I get it from the adrenal cortex. I know that. How's the adrenal cortex turned on? There's two different ways that I can actually get out, even if my water levels are perfect. If I have too much potassium, then that's going to cause aldosterone to be released, which is going to cause us to put potassium in the DCT, the distal convoluted tubule, to go out in the urine. In other words, I want to dump potassium to bring it down. And we've talked about the dangers of hyperkalemia, so yeah, it's kind of a good idea. Now, this is the part that we've kind of talked about. If your sodium levels are low, it's going to be detected by the macula densa, the juxtaglomerular apparatus. It's going to release renin, which is going to eventually turn into angiotensin II. Angiotensin II is going to do a lot of things, but one of the things it's going to do is cause the adrenal cortex to release aldosterone. The kidney tubules, we're going to do this. We're going to bring sodium out and put potassium in. And of course, chloride is going to follow. Now, if I bring sodium out, then water is going to follow. Water follows salt. So basically, at this point, I'm going to reestablish my sodium levels. Or if it's too much potassium, I'm going to reestablish them within the homeostatic ranges. This is pretty good. I've got another one on a later slide, but we'll do both. Aldosterone, we've already talked about why it gets released. We've actually talked about what it does. And here is kind of the pathway. Angiotensinogen is made by the liver. The kidneys, for whatever reason, is going to release renin. Renin is going to change angiotensinogen into its active form, angiotensin 1. Angiotensin 1 in the lungs, angiotensin converting enzyme, ACE, is going to convert it into angiotensinogen 2, or excuse me, angiotensin 2. Angiotensin 2 is going to go to the adrenal cortex. We've already seen about the aldosterone. It's also going to go to the posterior pituitary and cause the release of ADH to absorb water from the collecting ducts. It's also going to have a direct effect on the sympathetic nervous system, which is going to cause us to take our blood vessels and constrict them. So the vasoconstriction, by making them smaller, is going to increase blood pressure. Because if our sodium is low, then our blood pressure is probably low as well. We're probably hypotensive. Atrial natriuretic peptide. This is released by the atria of the heart. When the heart atria are subjected to too much pressure because of excess fluid, especially in the case of an injured heart, such as congestive heart failure, it's going to release this. This guy is going to go and basically shut down renin and all of this. It's going to actually cause the afferent and the efferent arterioles, the afferent to open up, the efferent to constrict, which is going to increase the glomerular filtration rate. That's going to cause more fluid to go into the kidney tubules or into the nephron. Natriuresis and diuresis. Diuresis is just increased urination. Natriuresis is increased sodium in the urination. By doing that, I'm going to decrease the blood volume, and that's going to decrease blood pressure. ANP will also cause vasodilation of those blood vessels. We're going from there to there, which is going to lower the blood pressure, which is what the heart needs. So the kidneys, is very important that they get enough blood pressure so that they can do their jobs, and it's extremely critical. However, it is more critical that you not cause your heart to completely fail. You're not going to survive that. So ANP is the trump card. If you've ever played Rook, I think the root card is the trunk in that, if I remember right. Okay, guys, here's another picture of the same thing. Angiotensin, the increased blood pressure due to vasoconstriction, increases GFR, constrict, and a little bit of dilation. The more I constrict the efferent arterio, the more blood I have or pushing through. Angiotensin will also, via the hypothalamus, increase send signals to the thirst center to make sure that we're thirsty and get our aldosterone release. We've already talked about all of this multiple times. Here's our EMP. I didn't tell you about BMP. Beta natriuretic peptide is released by the ventricles. Atrial natriuretic peptide by the atria. So increased stretch because of increased blood pressure. AMP is going to shut down the aldosterone release, going to shut down ADH release, going to shut down renin, and because of that, the angiotensin, which is going to lead to vasodilation, which is going to decrease our blood pressure. Without the release of renin, we're going to actually increase the sodium and water that's going out into the urine. By doing that, we're going to lower the amount of blood, of the fluid portion, which is going to decrease the blood pressure. So if we don't reabsorb as much water and sodium, we're going to have more going out in the urine. Okay. So we've talked about not enough or too much blood pressure. What happens when we have not enough? This is kind of complicated, so I'm going to try to go through this one little section at a time. So our blood pressure is low. How do we know? We got baroreceptors in the in the arch of the aorta and in the carotid arteries. So these things are going to report to the hypothalamus. The hypothalamus is going to control the sympathetic nervous system. The sympathetic nervous system is going to cause those arterioles to constrict, which is going to raise our blood pressure in the short term by increasing the peripheral resistance, which means making them smaller. Okay, by increasing that, that's going to increase the blood pressure. So there's this guy. So in the afferent arterio going into the kidney, if my blood pressure is lower, I'm not going to have as much stretch. Now, from the macula densa, that's part of the distal convoluted tubule, I'm going to have less flow of fluid, less sodium and chloride. These guys are both going to signal the granular cells in the juxtaglomerular apparatus to release renin. Renin, we've already seen, is going to cause angiotensin II. I'm not going to go through all that. Angiotensin II, we're going to get the same vasoconstriction, which is going to cause the blood pressure to go up. Okay, that's a temporary solution. We're also going to have the aldosterone, which is going to cause us to reabsorb sodium and water, which is then going to increase blood volume and bring our blood pressure back up. Angiotensin II is also going to posterior pituitary, ADH, to again increase the volume by increasing water reabsorption. It looks really complicated, but if you take one little piece at a time, it's really not that bad. Calcium and phosphate, PTH. Generally, what happens is if we reabsorb calcium from the kidney tubules, we throw phosphate in its place. It's kind of like the sodium potassium thing, only these guys have opposite charges. So PTH, calcitriol, which is vitamin D, calcitonin, no, nothing going on there. And you guys can go through this. We've already talked about this. PTH, break down calcium from the bones, stimulate uptake from the kidneys, and also activate vitamin D and increase calcium uptake, raising the blood levels of calcium. Acid-base balance. This is something we haven't talked about yet. So let's talk about this before I get started on, before I get started on that. Acidity is the number of hydrogen ions. We'll talk about pH on a different slide coming right up, but that's what acidity is about. If you have more than the neutral, then you're acid. If you have less than is normal, then you're alkaline. We'll talk about that more in detail coming right up. But this is all what acidity is about. This is very important for our homeostasis. We have to keep this under control or we can't survive. Slight pH changes have dramatic effects on things such as the enzymes, cell membrane receptors, hemoglobin, ion channels. What is the key common point? These guys are all left out. Proteins. Proteins have a certain shape in order to do their job. If that shape changes, then it's no longer any good, any more than if somebody took a file and filed down one of the ridges on my key to my car, it will no longer function. So this is what happens. pH can cause the enzyme or the proteins to change shape and make them non-functional. The sodium-potassium pump becomes 50% less when this guy changes by a factor of 10. If I have 10 times as many or 10 times fewer hydrogen ions, then I can have this guy go down 50%, which probably won't be much of a problem because you're really not going to know anything about it, because if you change your pH by 1, you will be dead. You draw a lily there. That's what's going to happen. So we know why we've got to be concerned about this. A great number of our ICU beds are taken up by people with erratic electrolyte imbalance and acid base. Trying to see if there's anything new on here. I don't think there is. Okay, here's our thing. 735 to 745. That's where we like to be. If this guy goes down from 735 to 7, you are dead. If this guy goes up 0.35 from 745 to 78, you are dead. DC is an extremely narrow range. Now from 735 to 7, you're in acidosis, which means we got to fix this or you're going down this way. Same thing with alkalosis, going this way. Too much hydrogen ions, not enough hydrogen ions. So decreased, increased. Okay, now it's time to talk about this. Turns out that pH is the negative logarithm of the hydrogen ion concentration of a solution. Did that go straight over your head? Okay. So pH of 7 is completely neutral. That's like pure water. Now a pH of 7 is actually 10 to the minus 7. I don't teach math, but just to let you know, 10 to the minus 7 is the same as 1 over 10 to the 7th. Okay? If I was more acidic, then I would be 1 over 10 to the 6th. Guys, the difference between this, I have 10 times more hydrogen ions here than I did here. How does that work out? The larger this number is, the smaller the fraction is. The smaller this is, the larger. Okay, 1 over 10 to the 1 versus 1 over 10 to the 2. 1 over 10 to the 2 is a smaller number. Why is that? 1 over 10 to the 1 is 1/10. 1 over 10 to the 2 is 1/100th. So that would be a dime and this would be a penny. Does that make sense to you guys? If not, think about it for a little bit. But the difference between each of these pH numbers, 7, 6, 5, 4, 3, 2, 1, the difference between 7 and 6 is 10. The difference between 6 and 5 is 10. The difference between 7 and 5 is 100. And actually, down at pH of 0 is like 10 million times more hydrogen ions than neutral. This stuff burns because there's a lot of hydrogen ions that are looking to get stable, and they'll be glad to tear your skin and everything up to make it happen. So you can see some of the representative substances for the acidity, and that's fine. So again, we said the larger the number under the fraction, the smaller the number. So 10 to the minus 8 has 1/10 as many hydrogen ions as normal. 10 to the minus 9 is 1/100th. 10 to the minus 14, 1 over 10 million. So normal has 10 million times more than this guy, because this guy is very severely depleted. I don't want to throw a monkey wrench, but if I have 1/10 as many hydrogen ions at pH 8, I have 10 times as many OH. So you could also say 10 to the 14th is 10 million times as many hydroxyl ions, OH. Whichever way it's easiest for you to grasp this. We're going to talk about this a little bit more in the following presentation. Hydrogen ions we're getting continuously. Carbonic and lactic acid by aerobic and anaerobic glucose metabolism. Sulfuric acid, we oxidize some of the sulfur-containing amino acids. Phosphoric acid, breaking down phosphoproteins, ribonucleotides. Acidic ketone bodies, breakdown of fats, and especially get a toxic accumulation in untreated diabetes mellitus. That's what DKA is, diabetic ketoacidosis. Acid versus base forming foods, basically meats, nuts, seeds, acidic vegetables, fruits are going to be the base forming. Okay. Buffer systems. I already showed you, so if I have too many hydrogen ions here, which means I'm acidic, then I already have a certain amount of bicarbonate. I think it's like, I'm trying to remember, it's like 23 milliequivalents. But these guys, if there's too many, these guys are going to go and make carbonic acid, and then they will break up the carbonic anhydrase into water and CO2. And you're going to breathe the CO2 out and keep the water. That's it. If you have too few, then we're going to take excess CO2 and water and turn it into bicarbon hydrogen ions. That's the whole point behind the buffers. We have a chemical buffer system. That's the bicarb. It's floating in your blood anyway. That's going to make rapid adjustments. So if you get a sudden surge of hydrogen ions, this is going to take out that and smooth it over. One of the things we do is check bicarbs on our patients' acidosis and alkalosis to see. And if we have too much bicarb, then you're probably alkalotic, because if you don't have enough hydrogen ions, you're going to make all this, and so your bicarbs are going to go up as you attempt to raise this. that's something you hear more about later. And of course, if we turn this back into CO2, then we breathe it out. That's the respiratory system. By blowing off CO2, we lower our acid levels because once we blow this off, this water cannot come back and turn back into those hydrogen ions. The renal system can adjust the renal system can adjust how much of the hydrogen ions that are secreted. Again, I can bring potassium out of here and put hydrogen ions in here. So I can actually do that swap out. And also, the kidneys, if you recall from the renal chapter, that's where we make bicarb. We can make bicarb using this same process. If you remember, we were doing that in the tubule cells. This is the slowest. This is very fast. This is the almost instantaneous. Okay, guys, buffer system. There's several different kinds. So they're dividing them into chemical and physiological. Bicarb, we've talked a lot about phosphate, the HPO4 could very easily turn hydrogen phosphate could easily become dihydrogen phosphate, or dihydrogen phosphate could easily become monohydrogen phosphate. Protein buffer system, we haven't talked about that in detail yet. We'll get to that. So these are our chemical buffer systems. Physiological is just a respiratory mechanism. Once we take this guy and turn him into carbonic and then break him down, we blow him off. And then the other is that renal mechanism of how we